Now that we have understood color blindness, hemophilia, X-linked disease, hypertrichosis, Y-linked disease, we understand how does sex determination take place in living organisms. So how does sex determination take place? In human beings, the chromosomal mechanism of sex determination is XXXY. In humans, the nucleus of each cell contains 46 chromosomes or 23 pairs of chromosomes. Of the 23 pairs of chromosomes, 22 pairs are autosomes responsible for determination of body characteristics. So, in a cell, we have 23 pairs of chromosomes, of which 22 pairs are responsible for body characteristics: color of hair, color of hair, color of skin, height, intelligence, heart, lungs, etc. And one pair is a pair of sex chromosomes. So 22 pairs are autosomes, and one pair is a pair of sex chromosomes. One pair is a pair of sex chromosomes responsible for determination of sex. You have sex chromosomes, namely X and the Y chromosome. X chromosome and the Y chromosome. And in males, two homomorphic sex chromosomes are XX. In males, two heteromorphic sex chromosomes are XY. So females are 46 chromosomes, 44 are autosomes, and XX are sex chromosome. In males, 46 are autosomes, 46 are chromosome, 44 are autosome, XY are the sex chromosomes. So how does it determine? Father is X and Y, mother is X and X. Gametes formed from the father are X and Y, from the mother X and X. If X and X fuse, it forms XX, a daughter. If X and X fuse, it forms XX and daughter. If X and Y fuse, it will form a son. If X and Y fuse, it will form a son. So 50% chance are daughters are formed with XX, 50% chance are sons are formed with XY. So it is the father's sperm which will determine whether it is going to be a son or whether it is going to be a daughter. Mother से तो X chromosome ही आ रहा है, and it is father's sperms which will decide whether X chromosome or Y chromosome. X गया तो daughter आ गया, Y गया तो son आ गया. So it is males who are heteromorphic. Males are heterogametic and produce two types of sperms. Females are homogametic and will produce only one type of egg containing X chromosome. Males are two types of sperms. One type of sperm has 22 autosome and one X chromosome, and the other type of sperm has 22 autosome and one Y chromosome. That is males, heterogametic. Females are homogametic, producing only one type of egg containing 22 autosomes and one X chromosome. This understands that sex of the child depends on the type of sperm fertilizing the egg, and is the father who is responsible for the sex of the child and not the mother. Mother is only going to give the X chromosome. So it is going to be the father's X or Y chromosome which will determine whether it will become a daughter or whether it will become a son. So fathers are heterogametic, males are heterogametic and determine the sex of the child. Now, so lots of medical technique like amniocentesis, ultrasonography, were developed to detect genetic deformities in females like Down syndrome that we had said. These are misused to determine the sex of the fetus. So, sex of the fetus, say amniocentesis, if you have X and Y chromosome, then that is a male that is seen by amniocentesis. If it is X and X chromosome, then one of the X chromosome functions as the main X chromosome, the other X chromosome attaches to the cell membrane and becomes a rounded body, which is called as the bar body. And अगर bar body दिखता है, then bar body indicates it's a double X condition, which means it's a female child. तो मेल होगा तो on amniocentesis we will not see the bar body क्योंकि एक्स एंड वाई क्रोमोसोम है. अगर एक्स एंड एक्स क्रोमोसोम है, एक एक्स क्रोमोसोम फंक्शनल रहेगा और दूसरा एक्स क्रोमोसोम बन जाएगा बार बॉडी, which indicates it's a female. So bar bodies are detected on amniocentesis and females are detected, and if it's a girl child with a bar body, it is aborted, which is a non-medical termination of pregnancy. This is because of a gross discrimination against the girl child. So, to prevent this, the Prenatal Diagnostic Techniques (PNDT) Act, Regulation and Prevention of Misuse Act 1996 has banned the use of medical techniques for sex determination. By amniocentesis and by ultrasonography, sex determination is possible, but we do not disclose the sex of the child to the parent. Sex determination is not allowed by amniocentesis and ultrasonography. It is not allowed. And we detect the sex ratio, which is the number of females per thousand males. And sex ratio is an important indicator of gender equality and status of women in the society. According to the 2001 census, sex ratio in India was 933 females per thousand males. So we have a low female sex ratio. So that indicates that more and more people preferred having the male child, and many female children were aborted. In Maharashtra, it was 945 in 1991 census, came down to 917 in the 2001 census. So there was a lot of discrimination against the female child. In Kerala, it was 963 in 2001 and now is 1026 females compared to 1,000 males. So sex ratio is favorable to females compared to males. And this is happening in Kerala because Kerala has 100% literacy rates. And wherever literacy rates are high, the discrimination against females is not there. And now we have a wonderful sex ratio of 1026 females per thousand males. That is in Kerala. But India, it's still bad. It's 933 females per thousand males. So this is sex determination in humans, which is XY males, XX females. Males are heterogametic and females are homogametic. Males produce X and Y, two types of sex chromosome. Female produce X and X, only one type of sex chromosome. This is in humans. So then we have sex determination in honeybees. Honeybees have something called haplodiploid sex determination. Haplodiploid. Parents may, females are diploid, 2n is 32. Males are haploid, n is 16. So males are haploid, n is 16. Females are diploid, 2n is 32. So females have two sets of chromosomes, males have one set of chromosomes. And females undergo meiosis to form N16 egg and N16 egg. And eggs are haploid. Males who are already haploid undergo mitosis to form N16 sperms. N16 sperms. And अगर N16 egg and N16 sperm fuse together, it forms 2n32, which becomes a diploid female. सो एक एग स्पर्म के साथ फ्यूज होता है and forms a diploid female, while one egg does not undergo fertilization. Without fertilization, it starts undergoing mitosis to form an N16 male. So males are formed without fertilization, and haploid males are formed from the haploid egg. That phenomenon is called as parthenogenesis. So इसे parthenogenesis कहते हैं. So अगर N16 egg N16 sperm के साथ fertilize होता है, so 2n32 females बनते हैं, which is a normal female being formed. और अगर N16 egg undergoes fertilization, then female, but without fertilization, it undergoes parthenogenesis to form N16 males. So यहाँ मेल का क्रोमोसोम नेक्स्ट मेल में जाता ही नहीं है. मेल का क्रोमोसोम नेक्स्ट मेल में जाता ही नहीं है. And male is formed exclusively from mother. So the male will have a resemblance to the mother because of her haploid egg, the next male was formed. So males do not form the next male. The males are formed exclusively from mother, so they have maternal characteristics in them. And that is the way a haplodiploid sex determination is found in honeybees. Then you have sex determination in birds. Birds में उल्टा होता है. You have ZW, ZZ or XYXX. So it is the females Z females are heterogametic with ZW or XY pair of sex chromosome. Males are homogametic and have ZZ sex chromosome. So in humans it was XXXY and males were heterogametic. In birds, it is XYXX or ZWZZ, where XY females are heterogametic and males are homogametic with ZZ sex chromosome. So humans are XYXX and humans are XXXY, birds are XYXX. Humans are females are XX, males are XY. In case of birds, it is females are XY, males are XX, written as ZW, ZZ. Sex determination in grasshoppers and bugs is XXX0. So males are having only one X chromosome, and males will produce two types of sperms, one with X chromosome, one with no sex chromosome. No sex chromosome. Females will produce X chromosome wala egg. If X fuses with X, it will form a female. If X fuses with no sex chromosome, it will become XO, it will become a male. So in grasshoppers and bugs, males are heterogametic with XO. In butterflies and moths, it's the other way around. Females are X0, males are XX. So females are heterogametic with one egg having X chromosome and one egg having no sex chromosome. And males are having X sperm. If X sperm fuses with X egg, it will form a male. If X sperm fuses with O, it will form a XO, that is a female. So चार काइंड के सेक्स डिटर्मिनेशन किए हमने. Humans are XXXY. As a matter of fact, humans and Drosophila, which is the fruit fly. Humans and Drosophila or the fruit fly are XXXY. Birds are XXXYXX. Birds are XYXX. And but grasshoppers and bugs are XXXO. And butterflies and moths are XOXX. So ये चार टाइप के सेक्स डिटर्मिनेशन किए. And honeybees are, males are haploid and females are diploid. So that is called haplodiploid type of sex determination found in honeybees. And it was Henking in 1891 who found the X chromosome. X chromosome was found by Henking in 1891. So, sir, we have analyzed the concept of sex determination in humans and other animals. I hope you have understood this.